DocTalk is brought to you by Merck Animal Health. At Merck Animal Health, we're shaping the future of animal health with pioneering science, connected technology, and insights-driven solutions to bring our customers an unparalleled portfolio of choices to improve cattle care and operational efficiency. We support you and your legacy by helping you meet the challenges of today with the innovations of tomorrow.
Folks, thanks for joining us today. Dr. Costa Cimiteris is going to join us. We're going to talk about things associated with embryo survivability and the cow and much, much more. Some great work that's going on here at Louisiana State University. Thanks for joining us. We'll be right back.
They're here, they're hungry, and they can't be stopped with ivermectin. Choose Safeguard when you deworm your cattle to take out resistant parasites like brown stomach worm, Cooperia, Nematodirus, and others. With Safeguard's efficacy, you can kill more resistant worms in your cattle, so you don't leave potential on the table. Consult your veterinarian for the diagnosis and treatment of parasitism. Then bite back at safeguardworks.com.
Hey folks, welcome to DocTalk. I'm Dr. Dan Thompson and I'm here with Dr. Costa Cimiteris. We're at Louisiana State University where Dr. Costa serves as an assistant professor here in the Department of Animal Science.
Yes, sir.
And his pedigree and his training is pretty remarkable. And we're lucky in the beef industry to get you maybe away from some of the biochemistry and human medical fields to come help us with some cow repro issues.
Well, thank you, first of all, for the invitation. It's a real honor to be on your great show. And thank you for the very generous introduction as well. I've been very lucky to have trained under some really great mentors. And I'm also very lucky to be in the field of animal sciences, studying early embryo development and maternal embryo communication, which I just think is fascinating from a sort of cell biology perspective, and also has the potential to help producers and the broader environment in the sort of One Health capacity as well.
You bet. Let's dive into kind of the first topic, which you're doing some research on, looking at pregnancy sustainability or survivability within the first few weeks.
Correct. So one of the things that my group focuses on is bovine conceptus elongation. So embryo, well, after fertilization, the embryo will develop to a blastocyst. That takes about 7 days. And then after that, the blastocyst will hatch. And about day 10, the embryo, now a conceptus, so the embryo and the extraembryonic membranes, will become a conceptus and start elongating. So it'll go from an ovoid to, from a spherical, to an ovoid to a tubular structure to a filamentous conceptus by about day 14, 16. And that process is fundamental for reproduction because the concentration of the maternal pregnancy recognition signal, interferon tau, that's secreted by the conceptus is proportional to conceptus size.
Okay.
So if the conceptus doesn't elongate enough, it won't secrete enough interferon tau, and the pregnancy won't be recognized, and so will be lost. So it's really, really important.
So let me make sure I got this. So we're getting out there at 7 to 14 days, and we're changing into what we would call an embryo or conceptus, right?
Correct.
And as that elongates in this tubular or filament type structure, it's sending out this hormone.
Yes.
To tell the cow, hey, I'm here.
Correct. Yep.
Attach, let me dock, right?
Yes, and don't recycle, don't go back into cyclicity.
Right.
So yeah.
Yeah, don't spit me out.
Correct.
Keep me here, dock me, and this is a point in time that we lose a lot of pregnancies in cows, correct?
Yeah, about 55% of pregnancies are lost before day 21. Yeah.
Yeah.
So it does coincide with a window of...
So when you're doing timed AI, when we're doing things that we really want this bull to match with this cow's genetics, this is a critical thing when we see probably some of these drops in AI rates.
Yeah,
Or ET. Yeah.
it's true. I mean, from a sort of more philosophical perspective, what's quite interesting about the uterus is that it serves a dual purpose in a way. On the one hand, you've got a biological pressure to procreate.
Yep.
And then on the other hand, there's a pressure to reject developmentally incompetent. So it's hard to say often if a pregnancy loss is in the biological sense good or not. I know in an agricultural sense, no pregnancy loss is good, right? But teasing out what makes a good embryo, what makes a bad embryo is really important.
Awesome. Well, we're going to take a break with Dr. Costa Cementeris here at Louisiana State University. We'll be right back.
Hello there, Dr. Nick Wagner, Professional Services Veterinarian, and today I wanted to talk about some tips for managing foot baths on your operation. Foot baths are an important piece of personnel movement on and off an operation, and foot baths are something that if they're not appropriately managed can be a concern. So one of the things with foot baths that you should do is try to make sure that you knock off or rinse off the organic matter from boots before you step in the disinfectant, and then allow the disinfectant to have the proper contact time for it to be effective. And then look at disinfectants that may stand up a little better with organic load, because again, if you're not consistently managing these, the organic load in them can cause them to not have the level of efficacy that you may want.
Thanks for watching Cattle Intel by Neogen.
When the ones who have your heart need your help, count on us for everything they deserve, delivered fast. We'll work hard for you so you can work hard for your dreams.
Hey folks, welcome back to DocTalk. Dr. Dan Thompson here with Dr. Costa Cementeris, where Dr. Cementeris is an assistant professor in the School of Animal Science here at Louisiana State University. He is a reproductive specialist, unbelievable training globally, and has come to a place where you have quite a history here at LSU in reproductive physiology.
Yes, sir. Yeah, one of the main attractions for me to move down here was the long and distinguished history in reproductive research at LSU and the facilities, and just the climate and the environment is very conducive to good research. And one of the, I guess, standout names from here is Dr. Robert or Bob Godke, who led some really pioneering work. And we now have a seminar series named in his honor here in the School of Animal Science.
And I assume that we built the resources and the laboratories and things that were advantageous for somebody in your background to come here. And I've just been completely impressed with the facilities, with the faculty, with the surrounding of, if you get a chance, Louisiana State is one of the top agricultural schools.
Well, thank you very much. And yeah, I agree. We have some great facilities, and we're very lucky. I'm very lucky to be here.
So let's go back to, we have an embryo that's elongating, and it's kicking out this hormone. And let's talk a little bit about, because some of your research is trying to figure out what that cow or what the uterus is saying back, correct?
Correct, yes.
So kind of walk me through that, elongation, and then what it is that you're trying to figure out on the cow side.
Okay, yeah. So there's a lot going on, right, during that period of time. And if you think that the embryo goes from, this very small, about 200 micron diameter sphere, to by day 20, this filamentous structure that measures about 20 centimeters long. This is really an incredible phenomenon from a cell biology perspective. So a really important day in bovine pregnancy is day 16. That's the day of maternal recognition of pregnancy, give or take. What that means is that gene expression changes in the uterus up until day 16 are independent of interferon tau. So day 16 is the day that the mother knows she's pregnant under normal circumstances, give or take. So that means that the cow has to prepare for conceptus elongation during every cycle until about day 16. And we know that conceptus elongation is dependent on uterine glands. So Dr. Tom Spencer's group had previously knocked out uterine glands in the sheep, and sheep and cow are very similar in terms of conceptus elongation, to the point that you can put a cow embryo in a sheep and it will elongate, right? So uterine gland derived substances are essential for conceptus elongation, and we're trying to figure out what are those substances that trigger that developmental process. So that's the sort of long and short.
And I think that it's important for all of us to recognize the time factor, right? Because as we have timed AI, as we do some of these things that are at our convenience, and we have cows up there and we're then transporting them to different pastures and summer turnout and that, you know, some of the things that could be going on in the body, whether it's cortisol or, you know, response to stress, there's the potential it could have an impact on uterine gland secretions.
Yeah, so yeah, heat stress, things like that have been extensively studied. And there's little doubt that the overall health of the animal has an impact on pregnancy, much like women, you know.
Yeah. So as we wrap up, just think about minimizing stress, minimizing you know, infectious agents, commingling, and things of that nature during this critical 16-day period. So thanks. We're going to take a break. When we come back, more with Dr. Sementeris. You're watching DocTalk, and we're glad you joined us.
In the livestock industry, castration is a common practice to ensure proper herd management. All methods are painful, regardless of the age of the animal. At Solvit, we could not ignore the clear industry need for better castration solutions. So we developed LidoBand, a novel lidocaine-impregnated elastrator, addressing the pain associated with band castration. It provides local anesthesia throughout the castration process. LidoBand, a small device that can make a big difference.
I've been a veterinarian since 1987. My wife and I set up a practice in Monette, Missouri in 1989. We started using therapies shortly after it came out. I was overwhelmed with the positive response that we were getting from producers. They could noticeably see a difference. I have to admit that I was pretty skeptical at first. I am currently 110% sold on the product. There is no doubt in my mind that this is an added product that we can use on these cattle that is going to make a significant difference.
Hey folks, welcome back to DocTalk. Dr. Dan Thompson here with Dr. Costa Sementeris. We're at Louisiana State University in Baton Rouge, where Dr. Sementeris is an assistant professor in animal science, reproductive physiology, reproductive science, and doing a lot of work with cows. And now we get into some of the kind of, I mean, what he's doing in the laboratory is not to be underrepresented, because he is trying to replicate things in vitro that we can test and learn more so that we can move to the cow and do some in vivo things, right?
Yeah. So my dream, right, is to achieve conceptus elongation in the lab, because right now it's not possible. The most successful attempts to extend embryo culture in the lab get to about day 11, and that's using a special culture medium. But past that sort of ovoid shape is very difficult to do in the lab, and so that's one of the things we're working on. We use different approaches to try to achieve that, and just to understand more about the determinants of conceptus survival. One of the approaches that we take to trying to figure out what does the embryo need to elongate is flushing the uterine fluid from cattle across different stages of the estrus cycle and analyzing that fluid, so metabolites, proteins, other substrates, and then seeing if the stuff that changes around day 10, if we add that in the lab, is that what the embryo needs to elongate? Another approach that we use is using organoids. So organoids are hollow ball of cells.
Okay.
So I'll rewind a bit. So how you make organoids, you take a biopsy from, in our case, the uterus, the endometrium.
Yep.
You digest that tissue,
mince it up
with enzymes, and resuspend it in a hydrogel, which is basically jello. And those cell clusters will spontaneously form these really nice hollow balls. And organoids have been derived from many different tissues. The most that I can recall interesting or cool was the snake venom gland organoids. So from snake venom glands, and then these organoids inside the organoid produce snake venom. They've been made from human and mouse tear ducts to produce crying organoids. And during my time in Missouri, we made them from human endometrial epithelial cells, uterine cells, and we found that they produce a substance that closely resembles uterine fluid. Okay. And here at LSU, we've made bovine endometrial epithelial organoids, and we are seeing whether that fluid produced by the organoids can help to extend embryo culture duration in vitro.
It's so, and we'll save kind of the reason and that as far as transferring an older embryo or more developed embryo. But folks, this is the kind of research we have to be supporting. These as you look at being able to study this in vitro, to be able to look at different substances, different therapies, different opportunities to improve reproductive efficiency of our cows, to first-time AI, to shorten your conception, your gestation or your breeding season. All of this is going to play into improved sustainability, genetics, reproductive performance, and profitability of your cow herd. So let's take a break, and then let's come back and kind of give the punchline of why this is important.
All right, thank you.
You're watching DocTalk, Dr. Costa Cimenteris. We're here at LSU. We'll be right back.
When it comes to treating BRD, you want a product that you can count on to get the job done at an affordable price. Macrosyn by Bimeda delivers on both. A straight-shooting, no BS tulathromycin that does what it's supposed to do. End of story. You don't need to take our word for it, though. Go to macrosyn.com for customer testimonials and head-to-head trial results. For your cattle and your bottom line, choose Macrosyn.
Time is money on the farm, and your cows are less productive when they're stressed. The Alertus on-farm test from IDEX allows you to quickly test cowside and identify open or pregnant cows within minutes on your schedule in the parlor, barn, or chute. It's more efficient for your farm, very simple to use, and puts you in control. With minimal training and reliable, fast results, sample-based pregnancy testing is better for beef and dairy producers. Learn more at idex.com/DocTalk.
When you spot BRD in your cattle, that's your golden opportunity to target infection and its associated fever with a single dose of Resflor Gold, the industry standard dual therapy. To learn more, talk to your Merck Animal Health rep or your vet and see label at resflorgold.com. Animals intended for human consumption must not be slaughtered within 38 days of treatment. This product is not approved for use in female dairy cattle 20 months of age or older, including dry dairy cows.
Hey folks, welcome back to DocTalk. Dr. Dan Thompson here with Dr. Costa Cimenteris. We're at Louisiana State University, where he is an assistant professor of reproductive physiology, reproductive science.
Yeah.
Technology science. I'll just say this, that incredible research group that he's established here, doing some great things for beef cattle reproductive physiology and conception rates, and has developed an in vitro model to help create this conceptus and elongation. What is the importance or the relevance of being able to do this in vitro?
Yeah, who cares? It's a great question. So the long-term vision of this work is to be able to transfer elongated conceptuses. So if you think, if you transfer an embryo,
Mm-hmm.
you are reducing the likelihood of pregnancy loss in that you are transferring a viable embryo in all likelihood, right, by transferring a blastocyst. But then there's still a huge amount of pregnancy loss after that. So if we can transfer, in theory, at day 14 conceptus, we can minimize pregnancy losses by quite a substantial margin.
And part of that is that day 16 is when we're having the communication.
Correct.
So the closer we can close that gap from, say, day 7 versus day 14, the survivability and the fertility should be improved.
Yeah, in theory, the later you transfer, the higher the likelihood of success, in theory. Now, there's a limit, and the limit is about day 20 because that's when implantation begins.
Okay.
You could argue that the limit's actually closer to day 16 because you need that interferon tau, although you could supplement that exogenously in theory as well. It would be a nice experiment to do if we could achieve conceptus elongation in the lab and see how far we can, what's the latest that you could transfer. But there's another, if you think anyone who's flushed at day 14 conceptus knows that they are incredibly fragile. They can break very easily. And in fact, when you flush them out, they're often quite knotted and tangled. And so I don't know from a technical standpoint how feasible it would be. There's also, you'd have to be able to cryopreserve them, which is, again, a question mark surrounding that. So it's not like if we can achieve conceptus elongation in the lab, then overnight we can transfer these. There's still work that would need to be done. But the rationale is that, the later we can transfer, the better the outcomes, because we know we're transferring viable embryos or conceptuses.
Yeah. So I think some take-home messages, right? There's a lot going on in that first couple of weeks after conception.
Yeah.
And minimizing stress, minimizing interactions, disease, whether it's commingling of cattle from different pastures, all of these, transportation, around that day 14, day 16, where that seems to be the real critical time, it just seems vital to fertility.
Yeah, and they're really all critical times. You need each previous step to achieve. So you have to have fertilization, right? You have to then achieve the blastocyst stage. You have to have elongation. You have to have maternal pregnancy recognition. So it's a domino effect, really. And conceptus elongation is just what we are zooming in on and trying to figure out.
It's extremely interesting. Thanks for being on the show.
Thank you very much. It's been a pleasure.
Yep. Thanks for watching DocTalk. Remember, if you want to find us on the web, you can find us at www.doctalktv.com. Always work with your local veterinarian. With Dr. Costa Cementeris, I'm Dr. Dan Thompson from Louisiana State University. We'll see you down the road.
At Merck Animal Health, we support you and your legacy by helping you meet the challenges of today with the innovations of tomorrow.
One animal has this huge impact on your operation.
BVDV is one of the most costly bovine diseases, with losses of $15 to $88 per head.
The virus will actually cross the placenta and get into the fetus. As it's developing, its immune system will recognize that virus as it's supposed to be here. And so then that's where our persistently infected animals come from. They're going to shed that virus their entire life, and then it's going to impact your operation.
It is important to work with your local veterinarian and partner with IDEXX for the best solution for your operation.